Hello everyone, welcome to 10-Minute Physiology. In today's video, I'm going to try to explain to you what facilitation and depression are in synaptic transmission. So let's give it a go. So here we have the synaptic terminal, and what I want to do is first remind you how neurotransmitters are released in the first place. So inside the synaptic terminal, we have neurotransmitters stored in vesicles. And these vesicles are going to be held at a specific region called the active zones. And the active zones are regions near the presynaptic membrane that have proteins that are responsible for the fusion of the vesicle. So at the active zones are where the proteins are that are responsible for fusing the vesicle with the presynaptic membrane and therefore releasing the neurotransmitters. So in order to release the neurotransmitters, we need a signal, and the signal is going to originate or start from the voltage-gated sodium channels. So as action potentials propagate down the axon, voltage-gated sodium channels open, allowing sodium to flow into the cell. The sodium will then depolarize the membrane, which will open voltage-gated calcium channels, which allows calcium to flow in, which will increase the intracellular calcium level, which will then facilitate the fusion of the vesicle with the presynaptic membrane, releasing neurotransmitters into the synapse. Now, the neurotransmitter that we're going to be looking at today is acetylcholine, and the synapse we're going to be looking at is the neuromuscular junction. Now, as a quick side note, before you go further into this video, if you don't know what a nicotinic receptor is, I highly advise you to go watch one of my previous videos where I discuss it in great detail. You can find it in my synaptic transmission playlist, the same playlist that this video is in. So if you don't know what it is, please go watch that first, because knowing what that receptor is critical in order to understand the contents of this video. So now what I would like to do is take a closer look at the muscle fiber. So here is the plasma membrane of the muscle fiber, also known as the sarcolemma, and inside the sarcolemma are nicotinic receptors. So nicotinic receptors are ligand-gated ion channels, which bind to two molecules of acetylcholine. And when these molecules of acetylcholine bind to it, it will open the channel and it will allow ions to flow through it. Now, the reversal potential for the nicotinic receptor, as you should know, is around 0 millivolts. And this means that when the cell membrane potential is below 0 millivolts, or more negative than 0 millivolts, and this channel is open, what's going to happen is that the current will flow through the open channel into the cell, depolarizing the cell. And when the cell membrane potential is above 0 millivolts and this channel is open, current will flow out of the cell through this channel and therefore hyperpolarize the cell. Now, if we bring in our acetylcholines into the synapse and they bind to the nicotinic receptor, it will cause the channel to open and it will allow current to flow through. Now, because the membrane potential of a muscle cell is far more negative than 0 millivolts, what happens is that the current will flow into the cell and therefore cause the cell to depolarize. Now, before we talk about what facilitation and depression actually are, it's incredibly important to understand the different types of vesicles that are present inside the presynaptic neuron. So let's take a look at the three different vesicles. So now what we're going to do is we're going to zoom in at the presynaptic neuron, and this is the presynaptic membrane. Now what we're going to be doing is looking at three different vesicle types, and these different vesicle types are going to be found at different distances away from the active zones. Now remember that the active zones are specialized regions where the proteins are located that are responsible for facilitating the fusion of the vesicle with the presynaptic membrane. So therefore, they are responsible for releasing neurotransmitters into the synapse. Now, each of these vesicles are going to be found at varying distances away from the active zones, which are the regions right near the presynaptic membrane. So the first vesicle is called the docked and filled vesicle. And docked and filled vesicles are vesicles that are filled with neurotransmitter, in this case acetylcholine, and are really close to the active zones. So in other words, these vesicles are really close to the proteins responsible for vesicular fusion, and therefore these vesicles are really easy to fuse with the membrane when calcium increases inside the cell. So therefore these neurotransmitters are the easiest to release. The second type of vesicle are called filled vesicles, and filled vesicles are vesicles that are filled with neurotransmitters but are far away from the proteins responsible for vesicular fusion. So therefore they're harder to fuse with the membrane. And the last vesicles are clear vesicles or empty vesicles, and these vesicles have no neurotransmitter in them. So as a general rule of thumb, as we increase the distance of a vesicle away from the active zones, the more difficult it is to release its contents into the synapse. So now that we understand the three different types of vesicles, let's now look at the experiment we're going to use in order to understand what facilitation and depression actually are. So I'm sorry for the wordy slide, but I want to use this wordy slide so that you have a really good understanding of what this experiment is doing. So in this experiment, we're going to use it to explain how facilitation and depression actually work at the neuromuscular junction. So how are we going to conduct this experiment? Well, in this experiment, we're going to have two cells. The first cell is going to be a motor neuron, and what we're going to do to this motor neuron is we're going to repeatedly stimulate it, and we're going to stimulate it at a frequency of 30 hertz. So in other words, we're going to repeatedly stimulate it, and we're going to do so at a frequency of 30 hertz. And now each stimulation that we do will be powerful enough in order to elicit an action potential. Now the stimulation will cause an action potential, which will propagate down the axon and cause the motor neuron to release acetylcholine. The acetylcholine will then diffuse across the synapse, and it will bind to nicotinic receptors on a muscle fiber. So the muscle fiber cell is going to be the second cell in our experiment. Now, as you know, when the acetylcholine binds to the nicotinic receptor, what's going to happen is that the receptor will open and it will cause the cell to depolarize. Now, the depolarization that is caused by acetylcholine binding to all of the receptors on a muscle fiber is called an end plate potential. Now, we can measure the strength of this end plate potential by having a voltage sensor attached to the sarcolemma. So that is what we are going to be measuring in this experiment. We're going to be measuring the amplitude or strength of the end plate potential with each sequential stimulation of the motor neuron. So this is a pictorial representation of the experiment, and this is the muscle fiber cell. So what's going to happen is that when we stimulate the motor neuron, acetylcholine will be released. The acetylcholine will bind to the nicotinic receptor, open the receptor, and allow current to flow in, which will depolarize the cell, causing an end plate potential. The voltage sensor will then sense the end plate potential, and it will translate the amplitude or the strength of this end plate potential to the monitor here. So it's important to note that the height of this line here corresponds to the strength of the end plate potential. The higher the line or the longer the line, the greater the amplitude or the strength of the end plate potential is. So now what we're going to do is we're going to look at the experiment itself, and we're going to use this experiment in order to understand what facilitation and depression actually are. So this is a close-up of the monitor, and on the y-axis we are going to be measuring the strength of the end plate potential in the muscle fiber in millivolts, and this red dashed line simply represents the threshold potential. Now the threshold potential for the muscle fiber is going to be the cell membrane potential at which the muscle fiber fires an action potential. And you don't have to worry about that too much in this video. We're going to talk more about that in a later one. But what I really want you to focus on is the amplitude of the end plate potential. So remember that we're going to repeatedly stimulate a neuron at a frequency of around 30 hertz, and each stimulation is going to be strong enough to elicit an action potential in the motor neuron. The motor neuron is going to release acetylcholine, which will bind to the nicotinic receptors. This will cause an end plate potential in the muscle fiber, and the magnitude of that end plate potential will be recorded on this diagram here. So let's begin the experiment. So let's stimulate our neuron once. So when we stimulate the neuron, what we see is that EPP forms, or an end plate potential forms. So the end plate potential, as you can see, is a depolarization as what we would expect. So let's stimulate it again. So when we stimulate the motor neuron again, what we see is that the second end plate potential is larger than the first. And when we do it again, we see that the third is larger than the second. And when we do it continually up to around 5 times, what we see is that there is a sequential buildup of end plate potentials. So in other words, with each sequential stimulation of the motor neuron, we get a greater end plate potential in the muscle fiber. And this process is termed facilitation. Now, it's okay if you don't understand this right yet. We're going to get more into it in more detail. But the first thing that I would like to talk about before we talk about how this facilitation is actually occurring is how the cell is going to manage calcium. So what we're doing is looking back in the presynaptic neuron, so inside the neuron, in the motor neuron. And remember that in order to release a vesicle, we have to have calcium. And remember that the calcium comes in through the voltage-gated calcium channels. So the voltage-gated calcium channels are going to open, which will increase the intracellular calcium level. And when the intracellular calcium level increases, the vesicle will fuse with the membrane and release neurotransmitters. Now, these vesicles are docked and filled vesicles. So in other words, these vesicles are really close to the active zones, and they are really quick to be fused with the membrane and therefore really quickly release their neurotransmitters. Now, we talked a lot about how calcium comes into the cell, but we haven't talked about how calcium is removed from the cell. And one way in which calcium can be removed from a cell is via the PMCA, or the plasma membrane calcium ATPase. And what this does is it pumps calcium out of the cell up its concentration gradient, so against its concentration gradient, by using the hydrolysis of ATP to power it. So the PMCA pumps out calcium using the energy of ATP. Now, from this diagram, you can see that we sort of have a balance of calcium coming into the cell and a balance of calcium coming out of the cell. And it's going to be the balance between these two opposing processes that explains how facilitation is occurring. So now that we know how calcium is mobilized and handled inside the synaptic terminal, let's now talk about it in terms of how facilitation actually works. So here is a synaptic terminal of the motor neuron that we are stimulating in our experiment. So now let's look at the data that we saw in the graph that we just saw on the monitor and correlate it to this diagram here. So let's do the first stimulation. So when we stimulate the motor neuron, the first thing that we'll see is an action potential propagating down the axon. Now, this action potential will depolarize the membrane, causing the opening of voltage-gated calcium channels. Now, let's just say that when we open the voltage-gated calcium channels, 4 calciums come in. This is just a random number that I came up with. So let's just say whenever we open the voltage-gated calcium channels in this hypothetical neuron, 4 calciums come in. Now, let's just say that all of these calciums are going to be used for the fusion of vesicles with the membrane. So let's just say that whenever we have 4 calciums come into the cell, 2 acetylcholines will be released. So in other words, there's a 2-to-1 ratio of calcium coming in and acetylcholine going out, respectively. Now, in order to stop the synaptic transmission process from occurring, we have to remove these calciums. And in order to do that, we bring in our PMCA. So the PMCA pump, let's just say, pumps 2 calciums at a time. So the PMCA pump will pump out 2 calciums first, and this will cause the intracellular calcium level to come down to 2 calciums. Now, before the PMCA pump can pump out the rest of the calcium from the intracellular fluid, another action potential comes because we stimulate the motor neuron again. So we stimulate the motor neuron again before the PMCA pump can pump out the rest of the calciums. So when the action potential comes in, it'll open voltage-gated calcium channels, which will allow 4 more calciums to come in, which will increase the intracellular calcium level to 6 calciums. Now, because of that 2-to-1 calcium coming in ratio to 1 acetylcholine coming out, what's going to happen is that 3 acetylcholines will come out this time. Now, take note that the amount of acetylcholines that are coming out from this stimulation is greater than the amount of acetylcholines that came out in the first stimulation. It increased by 1 acetylcholine. Now, remember that acetylcholine binds to the nicotinic receptor and causes an EPP in the muscle fiber. And since the second stimulation caused more acetylcholines to come out than the first one, the EPP, or end plate potential, generated by the second stimulation will be greater than the first. So, and this is because we have more calciums present inside the cell. Because the calciums that came in through the voltage-gated calcium channels have added to the calciums that were remaining inside the cell that have not been pumped out yet. Now, the PMCA pump will pump out 2 calciums at a time, and it does so again. So therefore, the intracellular calcium level will come down to 4 calciums. And then when we send in another action potential, 4 calciums will come in, increasing that intracellular calcium level to 8, causing 4 acetylcholines to come out, and the process keeps repeating. Now, in this table, I am summarizing the experiment of this hypothetical neuron on this table here in order to help you better understand what I'm saying, in case that didn't really make sense either. So, if you look at this table here, we start out with a synaptic terminal with 0 calciums in it. And when we do our first stimulation, an action potential comes in, and it causes 4 calciums to come into the synaptic terminal when the voltage-gated calcium channels open. So when the 4 calciums come in, this will cause 2 acetylcholines to come out, and this will cause a resulting EPP of this magnitude. So we get this magnitude of an end plate potential in the muscle fiber. And then the PMCA pumps out 2 calciums at a time, and before the PMCA pump can pump out all the calciums, another action potential comes in. So in this case, we start out at the second stimulation with 2 initial calciums. The action potential comes in, causes 4 calciums to come in, resulting in 6 total calciums inside the cell. This will cause 3 acetylcholines to come in, and an even larger end plate potential to result in the muscle fiber. So as you can see here, with each sequential stimulation, the amount of calcium inside the cell increases, and the resulting end plate potentials in the muscle fiber increase as well. So in other words, with each sequential stimulation, we are increasing the amount of calcium coming in, and as we basically store or build up more calcium, we are able to release more vesicles and therefore more acetylcholine. So therefore, with each stimulation, we have an increased amplitude of the end plate potential. So this is the summary of what I want you to get from this experiment for the facilitation part. So basically, whenever we stimulate a motor neuron repeatedly at a high frequency, this will basically cause the amount of calcium to come into the cell, the rate at which calcium comes into the cell, to be far faster than the rate at which calcium exits the cell. So whenever we are stimulating a motor neuron at a high frequency, the amount of calcium coming into the cell will far exceed the amount of calcium leaving the cell. This causes calcium to build up into the cell over time. So therefore, with each sequential stimulation, we release more acetylcholine per stimulation, and this causes the end plate potential of a muscle fiber to sequentially increase with each stimulation. And this is what the process of facilitation is in a nutshell. Now you may be asking yourself now, does this process of facilitation happen forever? And the answer is no. At a certain point, what you're going to see is that as we continually stimulate the motor neuron and cause the release of neurotransmitters, what we're going to see is that the end plate potential will gradually come down with each stimulation. And we'll see that it will gradually come down until it gets to a level a little bit above threshold. So I just put this break in here just to show many trials. So this part of the graph is called depression. So what is going on now? We were talking about only a few seconds ago that with each stimulation, the EPP increases. Why is it all of a sudden decreasing at this point? Well, in order to understand this, we have to bring in our vesicles again. So these vesicles here are the docked and filled vesicles. So these are the vesicles that are at the active zone and ready to be released. Now, in order to release these vesicles, we need calcium. So calcium comes in through the voltage-gated calcium channel, which increases the intracellular calcium level, which then facilitates the fusion of these vesicles with the presynaptic membrane, releasing neurotransmitters into the synapse. Now remember also that the calcium is removed from the inside of the cell via the PMCA pump, which uses ATP hydrolysis to pump calcium up its concentration gradient. Now, let's just look at what this neuron is going to look like at the beginning of the experiment. So at the beginning of the experiment, at 0 seconds, we're going to have all of our vesicles, all of our docked and filled vesicles ready at the active zone. And when the action potential comes in and opens the voltage-gated calcium channel at 0 plus X seconds, what's going to happen is that these docked and filled vesicles are going to start to be used up. So at this time right here, what we're going to see is we're going to have less vesicles present than we did before. And if we do another stimulation or further down in time, what we would see is that more vesicles would be gone. And as we further stimulate, what we see is that we increase the amount of vesicles lost. So over time, with repeated stimulations, we are gradually running out of docked and filled vesicles. So therefore, as we do repeated stimulations and we run out of these docked and filled vesicles that are ready to go, the amount of acetylcholine that's being released per action potential is going to be decreasing over time. So therefore, as we repeatedly stimulate the motor neuron, we use up most of these docked and ready vesicles, and therefore, as they're used up, we release less acetylcholine per stimulation. So therefore, the end plate potential is going to gradually decrease over time. So now let's summarize this video. In this video, we talked about that there are 3 types of vesicles in the synaptic terminal. The docked and filled vesicles are the vesicles that are close to the active zones, and they are the most readily released. The filled vesicles are vesicles that are filled with neurotransmitters but not docked, so therefore they're harder to release. And the empty vesicles have no neurotransmitter in it. We also talked about facilitation, and in a nutshell, facilitation is basically a process that occurs at high firing frequencies. It's basically a process in which calcium builds up inside the synaptic terminal, which basically causes you to increase the amount of neurotransmitter released per stimulation, resulting in a sequential buildup of the end plate potential. And then we talked about depression. Depression is a process that occurs at all frequencies in which the synaptic terminal is gradually depleted of docked and filled vesicles, causing the EPP to decrease over time. So I know this is confusing stuff and it's kind of hard to grasp, but I hope this video helped you. If you have any questions, please put them in the comments area. And in the next video that I'm going to post, I'm going to be talking about potentiation. And so I hope to see you there. So good luck on your studies. I hope to see you next time.